Our next speaker is Dr. Michael O'Sullivan from PathWest. Michael is a consultant clinical immunologist and immunopathologist at PathWest, Fiona Stanley and Perth Children's Hospital. And he's going to be talking to us today about the immunological response to COVID. So he is an investigator for the WA COVID-19 Immunity Collaborative Biobank project, in addition to all his other research interests, which I imagine he hasn't had very much time for in recent months. So thank you.
Thanks very much for the invitation to speak. What's now this afternoon, I think. So the title's slightly changed, I guess. It's more the immunologist's response to COVID-19 rather than our immunological response, and I think really nicely complements what Toby and Merrily have spoken about already. I'm presenting this on behalf of Dominic Mellon, who's the coordinating principal investigator and the sort of lead of the Wasik team. That may or may not be a photo of Dom, but last I heard from him, he was somewhere off Exmouth. So I get the pleasure of being here today. Thanks to Darren and the Department of Health through the FHRI, who've provided the infrastructure funding to establish the Wasik Biobank. I am employed by PathWest and PCH and Fiona Stanley, none of which relates to actually doing this work. So, I think early on, as immunologists, we understood that immunity to SARS-CoV-2 was going to be a really important part of our response. As we heard this morning, we've been really well protected in WA by the fantastic public health response and the collective effort that's gone into that. But the only way of really getting past that is to eradicate the virus or to provide immunity to the population. We've essentially eliminated transmission of the virus in Western Australia in the community. But we haven't achieved eradication, nor will we anytime soon. So I'm not immune to smallpox, doesn't matter, because no one's going to get smallpox. I am immune to measles, so if I go to Bali, doesn't matter. If someone comes back from Bali with measles, it doesn't matter. So elimination as a sustainable strategy relies on immunity. When there's a measles outbreak, it's the immunity of our population that protects it from becoming a pandemic. And we can then decide if we're more or less concerned about the top half or the bottom half of that front cover of the newspaper. If that was SARS, then that'd be on the top. As it is, I think we'd all be happy to relegate the top half of that newspaper cover to history. This is not SARS-CoV-1. In 6 months, we had an epidemic through sort of South Asia, Southeast Asia, that then disappeared. And there is no endemic transmission. So we didn't have to develop immunity to SARS because we never got exposed to it. Aside from the scale being about 10,000 times different on the daily cases on those two graphs, the graph on the right is what we're seeing with SARS-CoV-2. 18 months in and we're seeing new peaks in the number of cases worldwide. This is not disappearing anytime soon. So even with elimination, we need to understand immunity. So if we need to sort of fundamentally question what we're talking about with immunity, and there's potentially two components. Ideally, we want to protect everyone from getting infected, because if no one gets infected, no one can transmit the infection to anyone else, and no one's going to acquire disease. But failing that, we want to protect people from getting sick. And it's the second point that most vaccination studies rely on. They look for symptomatic COVID-19 disease as the primary outcome of an effective vaccine. And that's what we really rely on. If everyone didn't get sick, we'd be less concerned about the presence of the virus. But ideally, we'd like to also be able to achieve the first point. So the WA COVID-19 Immunity Collaborative, or Wasik, was formed in around April 2020, really as a kind of voluntary collaborative network of people who are interested in the question of immunity to SARS-CoV-2. It's made up of clinicians from a range of disciplines, diagnostic laboratory medicine specialists, clinical and basic science researchers, public health, which was a really important part of this, and consumers. And it was nominally divided into different work streams based on what we as immunologists felt would be important to understand relating to immunity to SARS-CoV-2. And that's the humoral immunity made up of B cells and antibodies, mucosal immunity being the surface where our body essentially first comes in contact with the virus, T-cell immunity, and immunogenetics that sort of underpin our immune responses. And we know from a lot of the research, and I won't go into much detail at all about the excellent work that's being done all around the world, but we know that different people respond differently after infection. So this is an excellent paper that followed up some patients from the US and looked at the breadth of their immune response, which occurred 1 month and 6 months after infection with SARS-CoV-2. And it looked at the important things in their immune system. So we know the B cells and the memory that your immune system has is important. We know the types of antibodies are important, and we know that the T cell responses are important. So if you're in the left-hand column as one of the black people here, you had everything switched on in your immune system a month later, which was about 60% of people. And then as the colours change and you get right down to these poor people in the bottom corner, they only had 2 of those 5 potential protective mechanisms switched on. Look back at the same group 5 months later, fewer black people, more white and grey people. The immune system changes its response and loses its memory, potentially at different rates in different people. So if we want to protect the entire population, they're not all one colour. We need to understand what's going on at an individual level. We don't really know what the nature of protective immunity is. We've got tests that we can measure, and we're learning more and more about the critical parts of the immune response that protect us against infection and disease. But that's something we're still learning, and it's certainly something we're still learning as a result of vaccines in different populations. We don't know how long immunity lasts for. We don't know if it's different following infections and following immunisation. And as Meryl's touched on, we don't know if it's different between someone who's healthy and someone who's sick. And it's the sick sort of patients in our community who are at most risk of disease. We don't have a test to tell someone if they're immune or protected from COVID. And we certainly don't have a test that can be routinely applied in a diagnostic lab at scale to diagnose immunity in the same way that we can diagnose infection. So the Wasik Biobank was formed as a biorepository to support basic science discovery, translational research and diagnostic assay development and validation that's relevant to SARS-CoV-2 immunity. So a pretty broad brief, I guess, not presuming that we actually knew what would be important. That's the job of the expert researchers in the community and the expert diagnostic laboratory scientists. As immunologists, we'll all have an opinion, but as we'd heard early on in the pandemic, everyone's got an opinion, and most of them are going to be wrong. So we really wanted to create the infrastructure that allows people to pose questions, test them rigorously, and then implement them into practice. Exactly the principles that sort of Toby had espoused earlier. The Wasik Biobank's been funded by the Department of Health through the FHRI. It's been supported by a wide range of investigators and their affiliated institutions and other programs that have been touched on in the last couple of talks. PathWest is the sponsor for this project, and the South Metro ATREC has been very generous in their assessment at short notice, including out of session for our applications. The Wasik Biobank will be a prospective cohort with 2 years of follow-up, and as Merily touched on, it's been approved by ethics and just pending governance approval to start recruitment. This is really going to follow on from the work that the COVID research response established in the biobank with the acute setting, and also the sort of forward-thinking nature of Toby's work to allow continued collection of samples from patients who've recovered from COVID. So it's under those ISARIC protocols and the COVID research response that we're currently able to continue following up the immunological response of these patients. And the thought being that the Wasik Biobank will take a kind of longer-term view away from the acute COVID setting, but also look at immunisation as well as infection. We're hoping to establish a biobank that allows researchers to compare the response of immunised people versus patients who've recovered from infection, and also look at the difference in immune response of someone who was a close contact and may have been exposed to the virus but not become sick, as well as other influenza-like illnesses. And I guess this is a sort of high-quality, high-intensity repository. So this might be where the initial discovery and validation happens, because we're looking at large volume samples from modest numbers of people to really optimise the tests in the laboratory setting, and then use the scale that can become by linking in clinical and laboratory data from large populations of people to really apply the value of those tests that are discovered through the biobank samples into a broader population to assess the clinical outcomes. So patients will give up to 800 mils of blood over the course of 2 years, 100 mils of blood at a time point, up to 15 vials of PBMCs, as well as saliva, plasma and granulocytes collected. It's about 1/5 of a blood donation, so it's a large volume of blood. unashamedly, but it's also really intended to provide a really high-quality laboratory resource for the researchers who are going to do the fundamental discovery work around the correlates of immunity. The sampling timeline, we've borrowed in the cases to align with much of what's been proposed by ASARIC. Now, the patients we will see at the moment will largely be recruited around this time point, having been convalescent cases returned from overseas. But should it be required in the future, we would have the opportunity to collect samples from acute patients. Obviously, someone who's sick in hospital is probably a little less amenable, as would their clinicians be, to giving up 100 mils of blood. And in the immunised cohort, we're really wanting to focus on understanding baseline, getting samples where we think the peak of the immune response will be, so about 3 weeks after your first dose of the vaccine, and then 1 and 4 weeks after the second dose of your vaccine, whatever schedule that might look like. And then we'll look to see whether those immune responses persist every 6 months into the future. So intensive sampling early when we think the greatest yield will be from examining immune responses, and then looking at long-term follow-up. And as Merrily touched on, I guess the intention was always to use this as a framework to expand into other cohorts of interest, and the most pressing of which from a clinician and patient perspective is immunocompromised patients. So a number of clinicians at very short notice have put their hand up to assist with the recruitment of patients from particular disciplines, focus being on patients who've received organ transplantation and bone marrow transplant, cancer, including those who are being treated with cancer immunotherapies, HIV infection, autoimmune diseases. We know these are the people at most risk of disease. We know these are people who will likely respond less to a range of other vaccinations. That's sort of in black and white in the immunisation handbook. We know some of these patients may be at more risk from certain vaccinations. So in collaboration with the Transform team, who will provide that repository of clinical data to understand these patients and understand their responses clinically to the vaccines, we've established an appendix to the Wasik core protocol, and that's the kind of flexible nature of this infrastructure, that at pretty short notice we were able to identify a cohort of interest, use the biobank infrastructure and protocols, attach an appendix with the rationale for that group, collaborate with a team collecting the clinical data, and then within 3 weeks of that proposal being up, it's been approved by ethics, and pending governance approval, we'd start that recruitment with a modification to the blood volumes, given these are patients in hospital. We've not missed the boat in understanding immunity. We're just starting to see some really nice papers that show that the amount of antibody that you've got that neutralises a virus after your different vaccine correlates with the degree of protection. Lots of antibody, almost 100% protection. And it's a very nice correlation, a group that the Kirby Institute in New South Wales had put together. This is the benefit of putting your talk together last night, is I got a tweet from someone I follow at the Garvan Institute who alerted me to this paper. It also infers that if you start with a less efficacious vaccine and you project the rate at which your neutralising antibodies might fall, your loss of protective immunity will vary greatly. Now, these are all hypothetical projections, but it tells us there's a lot we still don't know. And we certainly don't know within individual variability as to who'll follow that curve. So just getting everyone vaccinated is not the end of the story. We also are starting to learn the effect of treatment on vaccines, and this was a preprint from a team at the NIH. Small numbers of people, but it shows that prednisolone, which is probably the most widely used or misused immunosuppressant in clinical medicine in Australia, is second only to something that completely depletes all of your B cells in impairing your antibody response to an mRNA vaccine. The same mRNA vaccine that all of our patients less than 50 with chronic autoimmune disease and cancer are going to be getting. We need to understand whether these patients are protected and how we do it better. Small numbers, massive opportunity for us to contribute to this, but a really critical problem. Our vaccination strategy for these patients relies on an mRNA vaccine. So this is the governance of the Wasik Biobank, and obviously that's, as Merrily had touched on, something that's really critical to understand. I guess the benefit of this sitting within PathWest is in the way that WA Health Service providers have access to all of our clinical data as patients in the hospital anyway. PathWest is the sole public hospital laboratory provider, and therefore the samples can be governed and owned in the same way that all of the existing laboratory samples and data are. There's a scientific governance committee that will provide advice to Dominic as a coordinating principal investigator, And as Toby mentioned, it's really the biobank team who run the show day to day and make everything happen. Banks need to have deposits as well as withdrawals. This is supposed to be a dynamic biobank. It can evolve and adapt and expand over time. So we're very keen for people with any interest in research into SARS-CoV-2 immunity to approach us for access. As Peter Clinken touched on, we need to be a bit parochial. We want to build capacity and expertise in Western Australia. So we're keen to engage in international collaborations, but we want to build this expertise locally. We can't rely on someone in the US or UK or Sydney to give us the answers. So thanks to everyone who's been involved in this project. I'm sure I've forgotten some names, but the work that the ASARIC and COVID Research Response team has done was really critical to the evolution of the biobank. Merrill's Transform team, Jess and Sarah who are doing the day-to-day work, universities who have contributed some in-kind support and funding along with the Department of Health. And that's the Wasik email address. Questions, interest, just email and say, Michael told me to email. I'm not sure what I'm asking, but we'll put you on the newsletter mailing list. We'll give you updates when the access requests are available. Wasik at Health. Feel free just to drop us a line and we'll keep in touch. Thank you.
Thank you very much for another fantastic presentation. Your gift from the organising committee. And do we have any questions? Come on, we must have somebody in the audience who wants to ask a question. Yes, please come up to the microphone.
I was just wondering about, with your follow-up of the patients in the biobank, are you tracking them to see if they develop any illness throughout the follow-up, and would that change your protocol?
Yeah, so we will do a sort of patient-reported clinical assessment at the 6 monthly time points, but we're not getting detailed clinical data through Wasik. The benefit will be of linking in again eventually with Transform is even if the patients were recruited as healthy individuals, we'll be able to link back to know if they've come into a hospital. So absolutely that will change, I guess, the interest. An interesting group will be people who've had COVID and then get a vaccine. So we'll cross them over from being a kind of post-COVID case into an immunised group. But absolutely, I think the sampling and the follow-up will need to be adapted as we learn more about the impact of second infections or other infections over time.
Yeah, or people that get the vaccine and then develop mild COVID might be interesting as well.
Yeah, absolutely.
This is a purely sort of technical question. Has somebody provided you with a freezer farm for all these samples?
Yes, fortunately, so your colleagues at Murdoch and Alison Emery and the infrastructure at Perkins North as well. So we, the biobank team, are the people who coordinate it, and we've contracted in the expertise of laboratories who handle and process samples for a living and have very large buildings with lots of liquid nitrogen to store these for us. So we're not building a biobank, I guess we're just building the framework, and Murdoch and UWA have been excellent in that regard.
Thanks again.